my talk will be will consist in 3 parts. First of all, I will talk about general mechanism of photo transduction, just for your interest, because yes, everyone is interested how it functions. Then I will talk about structure of outer segment and data of transport of Opsin along outer segment and cones. And then I will talk about our computational model, what we particularly do here. So, yes, general question is whether Opsin can be transported along outer segment simply by diffusion. And the second question is, okay, if yes or if no, how different subcompartments of outer segment influences transport? Because we see that outer segment outer segment is a compartment. Yes, so we have several compartments of the cell: outer segment, inner segment, nucleus region, and a synaptic region. And one of the questions is how molecules are transported to these compartments, okay, or between those compartments. And here we ask how Opsin is transported between subcompartments of the outer compartment of the cell, okay? Because outer segment has very complex geometrical structure. Well, so general idea of photo transduction is as follows. When we have no light in dark conditions, visual neuron function in a very specialized way. So neuron is partly depolarized. Depolarized. Why it is so? Because there are several channels in the outer segment which are open for sodium and calcium ions, and there are channels in inner segment which are open for potassium ions, and so-called circulating current runs between inner segment and outer segment. So it flows along the cell, inside the cell and outside of the cell, forming such a loop. Okay, and because of this, the membrane potential is in every neuron potential is about minus 70 millivolt, and here in rods and cones it is about minus 30 millivolt, so it is lower in absolute value. Okay, and because of this, calcium ions enter synaptic region and cause glutamate release. So it is a very strange situation. In no light condition, neuron constantly releases glutamate and constantly acts to the bipolar cell. Okay? And when light is absorbed by the rhodopsin, rhodopsin activates G protein, which is in the inner part of the cell, and this G protein is called transducin, and transducin activates phosphodiesterase. And I must say that this is the example of G protein functioning, because we have multiple G proteins in every part of our cell, of our body, in every cell, and rhodopsin is a first example and most widely used example of G protein, because it is very easy to study, because it is in the membrane of photoreceptors in the eye, and it is activated by light, so it is convenient to study rhodopsin, much more convenient than other members of G proteins. And so here there is a general scheme of G protein function. So this is the first step of phototransduction, and I will briefly show you the propagation step. So after rhodopsin is activated, it activates transducer, and transducer activates phosphodiesterase, and phosphodiesterase hydrolyzes cyclic GMP. Cyclic GMP may be complex, but as it is, okay, cyclic GMP dissociates from open channels for sodium and calcium. This channel close. This channel close, so we have closed channels, and because ion exchanger works and constantly pumps maintains gradient of ions, so we have membrane potential decreased in absolute values. So cell becomes more hyperpolarized, and this causes stop of glutamate release in the synaptic region. So briefly speaking, what we obtain, absorbing light leads to the stop or partial decrease of release of glutamate, and this is the signal. Okay, so signal is decrease of the signal in synapse. Well, and in my work and in the future in this talk, I will not tell about this phototransduction mechanism. I will tell only about this protein, about rhodopsin. Okay. So I will go to the second part of this talk. So what we know about structure of outer segment and transport of rhodopsin in outer segment of cones. Outer segments in both rows and cones are constructed similarly, and that's why they are considered in parallel. So here I show you the structure of rod outer segment. It consists of multiple layers of membrane, and this membrane is organized into the discs, and this membrane contains rhodopsin. So rhodopsin is densely packed into the membrane of this disc. And most important feature is that, well, first of all, rhodopsin is really densely packed there, and second, these discs are separated from each other and from surrounding plasma membrane. So every disc is a separate structure, and rhodopsin cannot leave its disc and go to another disc. Okay. In cones, the situation from the one hand is similar. Membrane is also packed packed into the multi-layered structure, but these layers are not separated into disks. So all these layers are connected between each other and with plasma membrane which embraces these lamellas. So now it is not disks. Here is an error. It is lamellas. We call it lamellas, and we are interested in how rhodopsin is transported along this complex structure. Okay. So what we also have here, which is column between columns and rows, is that look here is connecting cilium. It is short neck between inner segment and outer segment. It is both cones, and here from inner segment to outer segment through connecting cilium we have an axonium. Axonium is really produced or constructed from microtubules. Okay, and people know that, well, there is axonium, and there are some molecular motors which go along this axonium. And the problem is that nobody knows now what these motors do. What do they carry actually? And so in general, rhodopsin can be transported from inner segment to outer segment and along outer segment, in particular in cones, but maybe also here in rows. So maybe it can be transported by these motor proteins in theory. But our calculation shows this it is very, very unlikely, because we have huge amount of rhodopsin in here, and it is very unlikely that this huge amount can be transported actively with using ATP. Actually, it is an energy-dependent process of transport of substance along microtubules. So there are two hypotheses that transport of rhodopsin is either diffusional or active, and here we consider the first hypothesis. We ask whether the transport of rhodopsin, in particular along outer segment of cones, whether this transport can be solely diffusional, fully diffusional. Okay. So what experimental data we have? We use photo recovery after photo bleaching approach. FRAP. It is widely used approach in biology, because when people invented green fluorescent protein, it become we now can fuse GFP, green fluorescent protein, with any or almost any protein in the body. So we use transgenic animals actually, and we can observe transport of these molecules in the microscope. So we use genetically modified frogs, and these frogs have a GFP either on rhodopsin or in syntoxin transmembrane domain. So they are shown here, rhodopsin with GFP and transmembrane domain of syntoxin with GFP. So this is a convenient simple molecule which we believe behaves diffusionally like rhodopsin. So it is called as a diffusional label. Okay, it cannot interact with any component of phototransduction cascade and cannot dimerize. As rhodopsin, people believe that rhodopsin can partly dimerize. So we use both opsin GFP labeled and syntoxin transmembrane GFP labeled in our experiments. And experiment consists in the following. So here we see outer segment of cone, and another part of the cell is somewhere to the left. And before the experiment, we see that it can, we see the fluorescent signal. So we excite at one wavelength and detected another wavelength. Then we bleach some small region in the cell, so it cannot now absorb light and produce fluorescence. And then during time, we see that the fluorescence signal and concentration, as we believe, becomes more uniform during time. Okay. So we collect three-dimensional stacks of such images. We plot some line along this three-dimensional structure, and we obtain profile of fluorescence before experiment and during time. Okay, and our aim is understand what we see here. Okay, and the problem is, first of all, is that this transport proceeds not along this line, but as I explained before, along these layers, which actually all of them are perpendicular to this line. So we cannot directly use some simple diffusion equation, okay? Because diffusional path is not in this line, but it is some complex part along this complex structure. This is the first difficulty. And second difficulty is that all the dimensions of the structure are below the resolution of optical microscopy. I will explain. We have so-called diffraction limit of optical microscopy. It is roughly half of the wavelength of light on which we detect something. So here it is about 0.25, 1/4 of micrometer. And for example, diameter of tip of outer segment is about 1 micrometer. Okay, so here I plot the so-called point spread function. Point spread function. So this is an ellipsoid of uncertainty. So we cannot resolve any 2 points which distance between which are below dimensions of this ellipsoid. Okay, and if we turn to not diameter but to distances between lamellas, the situation is worse because the distances between lamellas is about 10 nanometers, and they are much below the resolution of microscopy. Okay. So we cannot understand nothing, anything by simply viewing to this profiles. We need some modeling. Okay, so we need some model to understand these results. And now I will turn to the third part of my talk. If there are some questions, no, okay. So what we do, we know from experiments. For example, here I show you the representative experiment of how outer segment is organized. There are generally 4 subcompartments, so 4 kinds of membrane which differ between themselves. Okay, so first kind is a flat lamellar surface. So this is the ordinary surfaces of lamellas where most of rhodopsin stays. Okay, we note them LS here. Then this membrane turns in this part of the cell, and we call it U-turn. And another part of the cell where plasma membrane is located. Here we see plasma membrane that is very hardly recognizable because membrane is a very thin structure. Okay, so under the plasma membrane, membrane is organized, turns as a happy turn. So this is another kind of turn, and it can be viewed here. So it is here we see plasma membrane. These are flat surfaces. Here we have U-turns, and here we have hairpin turns. It also we have them here. Okay, so flat surface, plasma membrane, a hairpin turn, and U-turn. Okay, and we know roughly the geometry of plasma membrane. In particular, we know from experiments that the width of plasma membrane does not change along the outer segment. So plasma membrane is like a rectangular piece of membrane which partly envelop the column. Okay, and U-turn is proportional to the perimeter. And we also know molecules which organize hairpin turns. So hairpin turns are in rows and in columns, and they are organized by peripherin 2 molecules. They are shown red here. And U-turns are organized by chromium 1 molecules. So we know both geometry and molecular organization of these subcompartments, and we also know that in hairpin turns there is very low concentration of rhodopsin, actually about 0, and we know that in U-turn there is also low but non-zero concentration of rhodopsin. Okay, so we know geometry, we know molecular molecular bodies of the geometry, And we roughly know the concentration of rhodopsin in different parts of this structure. So we construct a mathematical model of this system. So we have three-dimensional structure. We have two-dimensional membrane in 3 dimensions. Okay, and here we see outer segment of cone. We construct only half of it because of symmetry condition. Okay, we use data on geometry of this membrane. We see here, plasma membrane is here, and hairpin turns are here under the plasma membrane, and U-turns are here in the region where there is no plasma membrane. Okay, so we generally reproduce organization of outer segment, to the best of our knowledge of the geometry and sizes of the structure. Okay, and then we use finite element modeling to solve diffusion equation for concentration of plasma membrane protein in time and space. So we solve mass conservation equation, which is shown here, with flux, which contains diffusional term and the term responsible for exclusion of rhodopsin or syntoxin from region where other molecules constitute U-turns. Okay, and in hairpin turn we actually do not solve equation for concentration because the concentration there is 0. We know it from experiment. So we solve this equation with some initial condition which correspond to the initial breach of outer segment in experiment. And the idea of this equation, despite that it has diffusional and exclusion terms, that we can reformulate it as a diffusion of actual concentration in spaces between excluded molecules. And in steady state, when flux is 0, this equation, diffusion equation, results in that concentration is inversely proportional to the available surface fraction along the surface. So the physical meaning of this equation that in steady state the concentration turns to the equilibration in the available surface between excluding molecules. So this is the task which we solve, and now I turn to some results. So when we solve this task, we understand how the flux along the surface is organized. Here are two examples for a real number of lamellas, 300 lamellas, and for small number of lamellas, just because it is easier to plot fluxes in the structure, and it is hard to plot a detailed structure here. So we see that flux is directed from regions well, it is all for initial time of 1 minute after the after bleaching. Okay, we see that flux is directed from regions of non-bleached regions to the bleached regions along the plasma membrane, and we see how it is organized in the so-called saddle points. So the molecules diffuse along the surfaces here to saddle point, and then they diffuse through saddle points and through plasma membrane, and then they diffuse again through another saddle point, points to the plasma, to the lamellar surface where there is no unbleached molecules. Okay, so we understand how flux is organized there on the membrane. And what's next? Yes, next we can study the sensitivity of different of the system to perturbation of diffusion coefficients in different parts of this membrane. What we do here, here we have profiles of concentration in lamellars, in flat lamellar surfaces. So this is time 0, and during time we have some equilibration, and this is the equilibrium concentration in time infinity. And we can observe, we can plot kinetics in the middle of this profile. It is plotted here, and we can characterize this kinetics by characteristic time. Okay, so the time, the characteristic time is the time when the difference between this value and infinite value, the final value drops 1 over e, or maybe here half time. Sorry, I don't remember. Maybe it is half time plotted. So we measure this half time, and then we can perturb diffusion coefficients in different parts of outer segment. And what we observe when we increase any diffusion coefficient, so the time becomes smaller, because with increasing any diffusion coefficient, the diffusion will be easier, more easy. If we decrease any diffusion coefficient, the time becomes high, because we make diffusion harder. But the value of this of this change is different for diffusion coefficients in different parts of outer segment. Okay. For example, here I don't really now see, because I have overlap of. I will shift it. Just a moment. Yes. So, for example, here we see that changes the diffusion in lamellars. It is black line. Changes time of equilibration very little. So diffusion in lamellars do not limit the equilibration. But the most drastic change is we change diffusion coefficient in plasma membrane. It is green curve. Okay, so this is it tells that diffusion in plasma membrane mainly limits equilibration in this situation. This is example for 100 lamels. So this situation is for not very long outer segment. And the next step is we can study the sensitivity in this region. So we can perceive diffusion coefficient not high. So here we increase them 3 times and decrease them maybe 10 times, I don't remember. But we can perceive them plus minus 10% in this region and see the slope of this lines, and so it will tell us the sensitivity of the system to these parameters in the normal range of parameters. Okay, and results of this sensitivity analysis is plotted here, and here we see, well, here in this axis we plot number of lamels. So this situation corresponds to 100 lamels, this one. And we now can use number of lamels as additional parameter when we study our system. Okay. So what we see here, that the sensitivity of equilibration time to the diffusion coefficient in lamelles is small for all number of lamelles. Okay, so if we increase or decrease the length of outer segment, the situation changes slightly. But the sensitivity of equilibration time to diffusion coefficient in plasma membrane, it decreases when we tend to normal length of outer segment. Okay, and the sensitivity of diffusion coefficient in U-turn, that is, U-turn is that is these structures, and so this is this part where lamelles and plasma membrane join. Okay, so the sensitivity of this part to the diffusion in this part decreases when we increase number of lamels. So this tells us that in a real situation, the transport of rhodopsin along plasma membrane of outer segment is mainly limited by diffusion along plasma membrane. So plasma membrane is a bottleneck to this process. Okay. And next, the problem is how to compare these results to experiments, because, well, this is a quite detailed model, but it is very heavy to solve. We have a huge number of computational nodes here. It is time of solution is quite high, and to compare to experiment, it is unfeasible now to do it. So we need some reduction, and we do this reduction. We reduce it. We reduce our three-dimensional model first to the two-dimensional model and then to one-dimensional model. And here I compared directly three-dimensional model to one-dimensional model. So here we consider diffusion along the one segment, along plasma membrane, and we consider the concentration. We suggest that concentration in lamelles is equilibrated along the radius of outer segment. Okay. So we solve here also the diffusion equation for concentration of syntoxin in membrane. So this is mass conservation equation, and this part corresponds to capacity of surface of plasma membrane and of lamelles in some coordinate x. Okay. So yes, and here also in the flux we see the factor of width of plasma membrane, which is constant, and diffusion coefficient, which is really not known in the system, and this is the only parameter which we don't know. So below, when we consider this model to experimental data, we vary this diffusion coefficient. Actually, we vary is ratio to the diffusion in lamelles, which is known. Okay, I will show you on the next slide. So yes, before we compare to experiment, we compare three-dimensional model to one-dimensional model, and here we see profiles of concentration in three-dimensional model and one-dimensional model at small number of lamelles, and they are not very close. But when we increase number of lamelles, when we tend to real length of outer segment, the situation becomes much better. Profiles becomes closer. The difference between them becomes less, and kinetics in center part becomes also closer. So here we see kinetics in three-dimensional model in the center part of the cone and in one-dimensional model. Okay, this for small number of lamelles, and this is for 300 lamelles. So they become closer and closer. Okay. Next, how we directly compare experiment to the model, or model to experiment. So we have profiles from experiment. We have dimension of particular outer segment. So we have diameter or radius here. Yes, here diameter, hypothetical diameter, which is measured directly for this cell. We have length of the cell. We have signal, fluorescent signal before experiment and during the experiment. Then we normalize the signal to this trend, and actually it is quite hard to do it because of it is hard to profiles. This is a live cell. Okay, this is not freeze cell, not frozen cell. It is live cell. It slightly moves. Okay, so there is processes. There are some processes. For example, dark current flows they inside the cells, and other processes around here. So really, we need to account for many experimental conditions, experimental factors which are there. I shown here about we need account for ballooning of the of the signal. So the difficulty is that in model we solve for concentration, but in experiment we see fluorescent signal. So we should recalculate from signal from concentration to fluorescence. Okay, we need account for anisotropy of point spread function, for tilt of the cell, so it is slightly tilted related to the play to the observation plate, okay, and so on. It's moving and bending. It bends slightly. You can see here. This is the same cell during time, and it bends and then restores. Okay, so we compare normalized fluorescence intensities to the calculated fluorescence in the model, and the only parameter is diffusion coefficient in plasma membrane. Here we have we see difference between experiment and model in every trace, and here we see that when we vary diffusion coefficient in plasma membrane, in some point we see we have the minimum of minimum error. Okay, we do it for multiple cells, the same approach, and we can compare this multiple data here in one slide. We can normalize, for example, here all the kinetics in the bleached minimum for to have the 10 to unit in infinity, and we see that after this normalization they become more close to each other. It was good for us, yes. So when the problem is that we cannot obtain infinity condition in experiment, but in model we can. Okay, so we normalize to initial condition in here and to infinity in experiment, and we have the spread between different cells becomes much less. Okay, so, and lastly we determine the diffusion coefficient in plasma membrane relative to the diffusion coefficient in lamelles, and this value actually different for different cells, but the average value is about 2, and actually it is the value which we expected before the start. So we expected that diffusion in plasma membrane is about 2 or maybe 2 to 5 times higher than in lamelles, and that's exactly what we obtained here. So I will skip the slide, and so general conclusion is that the answer is yes. So diffusion can work. The transport of rhodopsin between lamelles can work as a pure diffusional process. We do not need active transport to describe all this experiment, and moreover we obtain that the impact of control of different subcompartments is different. Plasma membrane controls about 80%, and cell point control about 20%, and impact of flat lamellar surface is about 5% lower. Okay. And we obtained the diffusion coefficient in plasma membrane, and it is about twice than we measured before in lamellar surface. And I want to acknowledge the lab where this experimental data are gathered. This is a lab of Professor Peter Coward in SUNY University in New York. So experimental data obtained by him. So that's all. Please, if you have any questions, you are very welcome.
Yes, thank you, Alexei. It was interesting. The topic was new for us, at least I think for many of us, in particular for me. Not always easy to follow, but we did also our best to understand what you present. So we have time to discuss. Please, if there are questions in the audience. And waiting for other questions, I will begin with my questions. So let us first of all try to understand what is happening in these experiments. You explained, of course, but again, since we are not very familiar with the subject, let us repeat. This bleaching experiment, in fact, how to understand that? Is my understanding correct that, for example, if we look at light in the dark, right? So during some time we lose our vision. Is it related to this experiment? So this bleaching reduces our visual capacity for some time, and after some time it restores. Is it about this in this experiment?
Well, actually, no. These are different bleaching. So what we see here in this experiment, this is a, well, physical bleaching of a particular molecule. So this GFP, it can do two processes. It can absorb light at one wavelength, and it can emit light. Okay, and this is called fluorescence. So what we do here, we pass two photons to this GFP, and it loses its ability to fluorescence. Okay, so it is the same as if we distract this molecule completely. So we simply exclude this molecule from the system, and we call it bleaching. Okay.
Oh, yes.
So when we look on the light, when we go from dark to the light, and they tell that the person is bleached, it is another bleaching. So we lose this ability to absorb light transiently, and this is the property of the phototransduction cascade, which I've shown you before in the very beginning of my talk.
Mm-hmm.
So we transiently, this cascade transiently cannot function properly, but then it restores.
Mm-hmm.
Okay.
Yes. All right. I asked this question, in fact, because I think that in the beginning you told us that rhodopsin is related to vision in the dark, and then this is why I thought that in the light, and in fact the experiment shows that the concentration of rhodopsin decreases, right, because of bleaching. So I thought that this is related. By the way, Okay, but I will return to this question a little bit later because we have another question. Angelica, you wrote that you have a question. Please go
Yes,
ahead.
thank you. I don't have that option to raise the hand. Thank you, Alexei, for this very interesting talk. If I understood, the diffusion coefficient is different in different lamellas. And regarding that, my question is the thickness of plasma membrane different in different lamellas? And if it yes, is it connected? Is there some kind of topology in the outer segment regarding that thickness? Because that could be affect the process of diffusion in that case.
Yeah. So let me tell it more clear. So we have, for example, here, we have different parts of membrane. We have lamellas, flat lamellar surfaces. We have here hepaton. We have plasma membrane. Okay, and well, uton is not shown here because it is for rows in particular. So the thickness of membrane is the same. So it is about 5 nanometers or about so, and it is the thickness. The thickness is due to the length of phospholipids which constitute this bilayer membrane. Okay, so you understand that membrane consists of phospholipids which have water.
Content of the phospholipids could differ in different lamellas and affect the diffusion coefficient.
So here the main effect on diffusion coefficient is from concentration of various membrane components other than lipids. Because, for example, here in hepaton we have lots of this protein. Okay, shown here, peripherin 1, and it is here peripherin is shown. Yes, this is red here in rods. Okay, so and besides from peripherins, there are another molecules with lower concentration, but the fact is due to this dense packaging of peripherin, there is no rhodopsin here at all. People cannot measure this concentration. It is believed to be close to zero. So if no concentration, then maybe no diffusion. Okay, so we cannot strictly say about diffusion, but it is, well, we can suggest that diffusion is hard, where if we place rhodopsin there, it will not diffuse because of dense package of great concentration of peripherin. Okay. It's the same for uton. Okay, so the main effect is from concentration of proteins. Then,
but there is some kind of topological pattern of distribution of that proteins. That's what I want to ask. Is there a pattern of distribution of that proteins?
Distribution of what?
Of different proteins. So see from these graphs is that like a peripherin is at the periphery, right? And there is no diffusion, and it's not uniform for the cones. I mean,
it is very good question, and we ask it for ourselves very frequently, and we can and we cannot measure. So we see the densities, but we cannot measure the special changes or special dependence of these densities. So I don't know.
Okay, maybe these guys from cellular biology, maybe they can do some measurement with fluorescence. Guys, I don't know.
Yeah,
but the problem is because you have a live cell, and you need the process in a live cell.
We have yes. So the question is that we well, one of the main questions here, one of the approaches, yes. So we tend to work in live cells because many information which obtained from freeze cells, for example, here, they
just the structure.
Yes,
this is just the structure, not the function, physiological function. It's there.
Yes. It's a structure, but I want to say that the dimensions of the structure measured from this freeze condition is believed to be altered by the experimental condition of obtaining the structure. So we need to, well, people try to recalculate these distances from measured in freeze condition to the live cells, and it is another difficulty which I didn't tell you about this in the report, but we spent lots of time in clarifying this information.
Thank you very much.
Then I will return to some other questions. In fact, it seems to me that I missed something in the very beginning, and in fact, why bleaching reduces the concentration of rhodopsin? If you can return to this question. I understand that it acts on these fluorescent molecules, but not on the rhodopsin molecules themselves.
Yeah.
But then why do we have diffusion? Because I don't see why bleaching changes the concentration of rhodopsin, but apparently it does change. Otherwise, you don't have diffusion. So please, could you clarify this?
Yes. So this is a very good question, and so bleaching actually does not change concentration of rhodopsin, but bleaching changes the concentration of rhodopsin which we see, which we can observe. So when we bleach, we split rhodopsin GFP molecules into two types. Okay, into bleached type, where GFP does not function, and non-bleached type. And plus to it, there is also native rhodopsin which do not have GFP. Okay, so overall, we have loss of rhodopsin without GFP, which is native molecule. We have some number of rhodopsin with GFP functioning, non-bleached, and we have rhodopsin with GFP, but GFP is bleached. And here we see the third type of molecule. We see the rhodopsin with GFP, non-bleached, and we don't see another two types of these molecules. but because of low concentration of this non-bleached GFP rhodopsin, we solve equation for this concentration.
Yeah. So
another conditions, for example, diffusion coefficient, which depend on the total concentration in some volume or in some place of the membrane. This condition do not change because when we bleach some molecules, total concentration does not change, so diffusion does not change.
Yes, so the total concentration is constant everywhere. It does not change because of bleaching, the total concentration. But if it take only like this bleached molecules, then their concentration change. And because of their like random motion of all of them, so effectively diffusion, so we see that this restoration of this image, right?
Yes.
All right. But then what is the purpose, in fact? I mean, what is the question which this experiment can answer? This is if the total concentration of rhodopsin remains the same. So why to measure that, in fact? Because physiologically we need this total, I suppose that we need the total concentration of rhodopsin. I mean, all the three types, in fact, and it does not change. So what is the purpose of this experiment?
Purpose is to understand the fundamental rules of transport. So the mechanisms of transport. So of course we do not want to change some physiology. Okay, so this is a model creature, frog, with model rhodopsin GFP labeled. And more than that, we can use substitute of rhodopsin transmembrane domain of syntoxin. This is simply a one helix cut from syntoxin molecule with GFP and observe its diffusion. So the purpose is very clear to understand fundamental mechanisms of transport in these membranes.
Mm-hmm. Assuming that if for some reason the concentration is changed, so how it will then be transported due to diffusion, for example, right? So it is not changed. It is not changed in this experiment, but it can be changed for some physiological reasons, right?
Yeah, if it will change, we have a dependence of diffusion on concentration, and we introduce it into the model as parameter. And actually we do it in another model, a more complex, which I not covered here, and so it is way as we do.
Well, I have in fact several other questions, but maybe I will just ask just the last one. It seems to be important for understanding what you present, the difference between rods and cones, because as you explained in cones, lamellas are connected to each other and by this outer membrane, and then this diffusion in this outer membrane compensate eventual loss of rhodopsin. But in rods, they are not connected. You explained in the beginning that they are isolated from each other. So the same diffusion mechanism will not work in this case, right?
Yes. And actually, there is no any transport of rhodopsin between discs in rods.
So they're not. But the experiment which you presented, it's only in cones. So in rods,
yes,
it wasn't done in rods. This experiment.
It was done, of course, many times, many dozens, maybe in 70s, in 74 and 76, in previous century, and there is no transport of rhodopsin along road.
So if you show the same experiment as you did, so the concentration will not return to its original.
Yes, it will.
So it will stay. It will stay like that, right?
Yes. Yes. Exactly.
Yes. Okay. I see. Yes, this is indeed very, very interesting.
Moreover, the piece of data which I didn't show, there is constant formation of new disks here, and they push this disk upwards. Okay, and there are another cells here. Actually, it is retinal pigment epithelium cells which constantly harvest the tip of outer segment. So there is constant flux of this disks upwards, and people saw it long before. We can label rhodopsin with radioactive labels with amino acids, for example, and there is layers of these amino acids which go from base of outer segment to tip of outer segment. It is well known, and for cones it does not. So here concentration is diffusion along outer segment, and this also proves this idea of difference between rods and cones.
So if you return to this image with this experimental distribution, yes, this one. So in rods, this dark part will move. It will not become light again, but it will move to one side, right? Because of this, as you explained, there are new disks which appear and they push all that.
Well, this particular profile will not move because of length of time scales of these processes. Because here the experiment is on the scale of minutes and tens of minutes, but the replacement of these disks is about 10% in day in frogs. So about a week they need to replace the whole disk. So it is smaller, but it is very, very well transport effective process. There are lots of rhodopsin which is constantly transported here, and not only rhodopsin, also membrane components, phospholipids, which are constantly transported here and constantly harvested by pigment epithelium cells from the tip, and every cell maintains this elevator, maintains this ladder every day.
And also, well, according to your results and your explanation and cons, active transport through microtubules is not essential, but maybe it is essential for rods, right?
For rods, it is also very, we believe that this is not essential because the number of molecules which should be transported, for example, here through connecting cilium is about 1,000 in second. So it is really huge number of molecules, and it is very unlikely. Well, we cannot explain how it can be transported by active transport. So very, very unlikely. And another argument is that really rhodopsin is transported from inner segment to connecting cilium via these vesicles, and there is no data how these vesicles fuse with membrane, but we understand that they cannot go through connecting serum simply geometrically. So really it is a mystery, how rhodopsin is transported from inner segment to outer segment. And this is the focus of our another study, but I didn't talk about this data today because we have no time, but we work on it.
All right, very good. So now we are prepared for the next part, for your next talk, when you understand, when you solve this mystery, how it works in rods. We are ready to listen. So please tell us. It will be interesting to know how it works. Thank you very much. There are no other questions in the audience. So it was an interesting talk about the subject which we are not very familiar with. Thanks again, and we finish our seminar.
Thank you very much.
Yes, have a good day. Goodbye to everybody.
Thank
Bye-bye.
you very much. Goodbye.
Bye-bye.
Goodbye.
Bye.